So the astronomer Carl Sagan, uh, he famously pointed out that the nitrogen in our DNA, the calcium in our teeth, the iron in our blood, and the carbon in our apple pies were all made in the interiors of collapsing stars.
Right. We are made of star stuff.
Exactly. We are quite literally made of star stuff. And it's this brilliant, really poetic thought that just makes you feel kind of infinite. But to set the tone for this deep dive, I want to juxtapose Sagan with a much more blunt, grounded definition from Mike Adams. He said, and I quote, "Biochemistry is the study of carbon compounds that crawl."
Yeah, and that tension, that's exactly where the reality of human biology lives. I mean, we are assembled from infinite cosmic dust, sure, but right now, in this exact moment, those atoms are organized into these highly regulated, squishy bags of chemical reactions.
Squishy bags. I love that.
It's true. We're desperately trying to extract electrons from, like, the bagel you had for breakfast just to maintain structural integrity. You know, the poetry only exists because of a relentless mechanical grind at the microscopic level.
And that gritty, mechanical reality is exactly our mission today. We are welcoming you to this deep dive into the invisible microscopic universe happening inside your own body right now. We have a massive stack of medical and scientific notes covering the foundations of human life.
That's a lot of ground to cover.
It really is. But to actually understand these systems, you kind of have to discard the high school biology models.
Right.
We are not just talking about simple diagrams or dominant and recessive Punnett squares here.
No, definitely not. We're getting into the actual spatial geometry, the electrostatic physics of it all. Because understanding these systems isn't just about memorizing pathways. It's about seeing the beautiful, kind of terrifying logic of how humans are built and fueled.
So let's start at the absolute foundation, the molecular architecture, the blueprint itself, DNA. Now, we know the genome contains billions of base pairs, and the notes say if you stretched out the DNA in just a single one of your cells, it would measure roughly 2 meters long.
Which is incredible when you think about it.
Right, because it has to fit into a nucleus that is only about 6 micrometers in diameter. So this isn't just a storage problem. It's an extreme electrostatic packing problem.
It's an engineering marvel, really.
Yeah.
Because DNA is fundamentally an acid, deoxyribonucleic acid. And the backbone is made of these phosphate groups, which carry a very strong negative charge.
Okay, so they repel each other.
Exactly. If you just shoved 2 meters of negatively charged DNA into a tiny microscopic sphere, the electrostatic repulsion would literally tear the nucleus apart.
Wow. So how does it not just explode?
Well, to solve this, the cell uses these proteins called histones. And histones are incredibly rich in specific amino acids, lysine and arginine, which carry a positive charge at a physiological pH.
Okay, let's unpack this. So it's essentially a magnetic neutralization.
Precisely.
The negative phosphate backbone of the DNA tightly wraps around these positively charged histones, like thread around a spool.
That's a perfect analogy. And this complex of DNA and histone proteins together, that's what we call chromatin. But what's fascinating here is that the spatial geometry of this chromatin, how tightly it's packed, actually determines the destiny of the cell.
Because it's not all packed the exact same way, right? The genome has like 2 primary states.
Right. First, you have your chromatin. This is the loosely packed DNA. I like to think of it as the open reading room of a library. Because it is physically open, the transcription machinery, the enzymes that read the code to make RNA, can actually dock onto the sequences. So euchromatin is transcriptionally active.
But the cell doesn't need to read every gene all the time. Like a neuron in your brain doesn't need to produce liver enzymes.
Exactly. That would be a huge waste of energy. So the cell takes the unnecessary sections of the blueprint and tightly coils them into the second state, which is heterochromatin.
The restricted archives of the library.
Yes. Heterochromatin is highly condensed. In fact, if you look at a nucleus under an electron microscope, the heterochromatin literally appears as darker, dense patches, usually pushed out toward the edges of the nucleus.
And because it's so tightly packed, the machinery just can't get in there.
Right. It is sterically inaccessible. The massive protein complexes required for transcription physically cannot squeeze into that coiled structure. The genes are effectively silenced by physical occlusion.
Which brings up a really crucial mechanism.
Yeah.
Epigenetics. Because the cell has to dynamically switch sections of DNA between these open and closed states, but it does it without changing the core sequence of the A, C, T, and G letters.
Yes, and the primary way it achieves this is through molecular on and off switches, specifically methylation and acetylation.
Okay, let's look at DNA methylation first. Mechanistically, how does adding a methyl group, which is just a carbon and 3 hydrogens, how does that actually silence a gene?
Well, it acts on a couple of levels. Typically, an enzyme called a DNA methyltransferase comes in and attaches a methyl group directly to the cytosine nucleotides, usually in areas known as CPG islands, which are near the start of genes.
Right.
First, just the physical presence of that bulky methyl group sticking out into the groove of the DNA double helix directly blocks transcription factors from binding. It's literally like putting a padlock on a zipper.
So it's physical interference right off the bat.
Exactly, steric hindrance. But second, and honestly more powerfully, that methyl group acts as a chemical beacon. It recruits specialized proteins that act like foremen on a construction site.
Okay, what do these foremen do?
They recruit massive chromatin remodeling complexes, including things called histone deacetylases, and these complexes strip away acetyl groups from nearby histones, which causes the entire local region to tightly condense into that dark heterochromatin we talked about. So methylation basically initiates a localized collapse of the chromatin structure. DNA methylation makes DNA mute.
Mute. Got it. And the clinical implications of this are massive. I mean, the notes mention X chromosome inactivation. So in individuals with two X chromosomes, one of them is almost entirely shut down, right?
Right, it's condensed into a tight, highly methylated ball of heterochromatin called a bar body.
Why does it do that?
It's a dosage compensation mechanism. If both X chromosomes were fully active, there would be this toxic overproduction of all the proteins coded on that chromosome. But dysregulated methylation in general is highly pathogenic.
Like when it happens by accident.
Exactly. Take Fragile X syndrome, for instance. It involves a massive expansion of repetitive code in the FMR1 gene. The body recognizes this abnormal expansion and aggressively methylates the entire region.
Oh, wow. So it just locks the whole thing down.
Completely silences the gene, and that leads to profound intellectual disability. The genetic code is technically there, but it is permanently locked away in the archives.
That's incredible. So if methylation is the padlock, acetylation must be the key. You mentioned histone deacetylases earlier, but the reverse process, histone acetylation, is how the cell opens those archives back up.
Yes. Remember that the tightly packed chromatin is held together by the magnetic attraction between the positively charged histones and the negatively charged DNA.
Right.
So an enzyme called a histone acetyltransferase comes in and covalently bonds an acetyl group to that positive histone tail.
Which chemically neutralizes the positive charge.
Exactly. The positive charge just vanishes. The magnetic attraction to the DNA backbone is broken. And so electrostatic repulsion takes over, the chromatin fiber physically relaxes, and it unravels from dense heterochromatin into open euchromatin.
And transcription skyrockets. Histone acetylation makes DNA active.
Yes. This continuous dynamic balancing act, methyltransferase is hiding the code, acetyltransferase is exposing it. That is the literal foundation of how a cell knows what kind of cell to be.
Okay, so let's drill down even further. Past the histones, past the packing, down to the actual letters of the code, the rungs of the DNA ladder.
The base pairs.
Right. We have the purines, adenine and guanine, pairing with the pyrimidines, cytosine, and the thymine. And there's a mnemonic in the notes for this. Purines have two rings, pure as gold. Adenine, guanine. Pyrimidines have one ring, CUT the pyramid. Cytosine, uracil, thymine.
That's a classic way to remember it. And we know A pairs with T and C pairs with G. But there's a really fascinating thermodynamic difference between these bonds.
Yeah, the notes highlight the hydrogen bonding. When adenine pairs with thymine across the double helix, they share 2 hydrogen bonds. But when cytosine pairs with guanine, they form 3 hydrogen bonds, like crazy glue.
Exactly. 3 bonds are physically stronger than 2. So if you have a segment of DNA with a massively high CG content, it literally requires more thermal energy to separate the strands. It has a higher melting temperature.
Wait, melting DNA?
Yeah. And this isn't just laboratory trivia. It has profound evolutionary implications. Think about extremophiles like the bacteria that live in boiling hot springs or deep-sea hydrothermal vents.
Oh, because the heat would normally destroy their DNA.
Right. Their environments would normally denature and melt standard DNA apart. To survive, their genomes have evolved to be extremely rich in CG base pairs. They rely on that third hydrogen bond to prevent their genetic code from literally boiling apart.
That perfectly illustrates the sheer physical fragility of the molecule. I mean, the genome is under constant assault. The text specifically highlights ultraviolet radiation. Every time you walk outside, UV light is penetrating your epidermal cells and physically warping your DNA.
UV radiation carries enough physical energy to break the bonds within the DNA molecule itself. Specifically, it causes adjacent pyrimidines on the same strand, usually two thymines sitting next to each other, to improperly bond directly to each other.
Instead of reaching across the helix to hold hands with an adenine.
Exactly. They hold hands with each other, creating what's called a cyclobutane pyrimidine dimer. It creates a massive, bulky kink in the DNA backbone.
And that kink is a huge problem when the cell tries to copy the DNA, right?
It's catastrophic for DNA replication. When the DNA polymerase enzyme, the copier, hits that bulky dimer, it stalls. And frequently it gets confused and inserts the wrong bases, leading to mutations and eventually things like melanoma.
But obviously we don't all instantly develop skin cancer every time we go into the sun. The body must have a repair mechanism.
It does. It has a dedicated crew called the nucleotide excision repair pathway. These complex proteins constantly scan the genome, but they aren't reading the letters. They are physically feeling the three-dimensional structure of the double helix.
Like running your hand along a rope to find a fray.
Great analogy. When they detect the physical bulge of that thymine dimer, they deploy endonucleases. These enzymes act like molecular scissors, and they don't just snip out the two bad bases, they cut the DNA backbone several nucleotides upstream and downstream of the lesion.
Okay, so it's like cutting out an entire section of rotted drywall rather than just painting over it.
Exactly. They remove the entire damaged segment, and then a fresh DNA polymerase fills the gap using the undamaged complementary strand as a template.
What happens when that specific scissor machinery is defective?
The clinical result is a devastating disease called xeroderma pigmentosum. Patients with this autosomal recessive condition have inherited mutations in the genes coding for those exact nucleotide excision repair enzymes.
So they completely lack the ability to repair UV damage.
None at all. From a very young age, even minimal exposure to sunlight causes severe blistering, extreme photosensitivity, and an astronomically high risk of developing aggressive skin cancers. They basically have to live their entire lives shielded from ultraviolet light.
Wow. And that is just dealing with external damage. But the replication machinery itself makes inherent errors, right? The notes detail the mismatch repair system, which operates right behind the replication fork.
Yeah, DNA polymerase is incredibly accurate, but it operates at staggering speeds. Occasionally, it inserts the wrong nucleotide, a mismatch that evades its own initial proofreading. The mismatch repair system identifies these unpaired, bulging regions.
Wait, let me ask you this. If there is a mismatch, say, a G is accidentally paired with a T, how does the repair system know which strand is the original correct template and which strand is the new one with the error? Because if it guesses wrong, it cements a permanent mutation into the genome.
That is a brilliant mechanical question. In bacteria, it's easy. The old template strand is heavily methylated over time, while the newly synthesized strand takes a few minutes to acquire its methyl tags. The repair machinery just targets the unmethylated strand.
But what about humans?
In humans, the mechanism is slightly different. It involves recognizing single-strand breaks, or nicks, that are naturally present in the newly synthesized lagging strand. The machinery assumes the strand with the nicks is the new one.
That's so clever.
It is. But if these mismatch repair genes are mutated, the error rate during cellular division skyrockets.
Which leads to Lynch syndrome, right?
Also known as hereditary non-polyposis colorectal cancer. Without mismatch repair, mutations rapidly accumulate, leading to a drastically increased risk of colorectal, endometrial, and ovarian cancers, usually at unusually young ages.
So we've talked a lot about how errors occur. But let's look at the functional impact of those mutations. Because a mutation is just a change in the nucleotide sequence. Are all mutations fundamentally bad?
Not at all. It's a spectrum. The severity is dictated by the ultimate effect on the three-dimensional folding of the resulting protein. The least impactful is a silent mutation.
And that's because of the wobble in the genetic code.
Yes. The genetic code is degenerate. There are 64 possible three-letter codons, but only 20 amino acids. Therefore, multiple codons code for the exact same amino acid. Often, changing the third nucleotide in a codon, that wobble position, doesn't change the amino acid that gets incorporated.
So the sequence of the DNA is altered, but the final protein is exactly the same.
Structurally identical, hence silent.
Okay, moving up the severity scale, we hit missense mutations. This is where the single letter change actually does alter the amino acid, and the clinical example here is universally taught. Sickle cell disease.
Sickle cell is the quintessential example of how a microscopic chemical alteration dictates macroscopic pathology. It's caused by a single point mutation in the beta-globin gene. The codon, JAGE, which codes for glutamic acid, is mutated to GTG, which codes for valine.
And glutamic acid is hydrophilic. It likes water, but valine is hydrophobic. It repels water.
Precisely. And remember, hemoglobin exists in a highly aqueous environment inside the red blood cell. Normally, that hydrophilic glutamic acid sits happily on the exterior surface of the hemoglobin molecule, but in sickle cell, you now have a hydrophobic valine on the surface.
Oh, I see where this is going.
When the hemoglobin releases its oxygen, it changes shape slightly, exposing this hydrophobic valine. To escape the surrounding water, the valine patch physically binds to a hydrophobic pocket on an adjacent hemoglobin molecule.
They start sticking together.
They polymerize. Billions of free-floating hemoglobin molecules rapidly lock together into rigid, insoluble, microscopic rods inside the cell. And these rods physically stretch and distort the red blood cell into that characteristic crescent or sickle shape.
And those rigid cells can't navigate the tiny capillaries.
Right. They create log jams, they cause excruciating vaso-occlusive crises and ischemic organ damage. And it's all because one single amino acid desperately wanted to avoid water.
That just perfectly illustrates the extreme fragility of our biological machinery. We rely on atomic-level hydrophobicity just to safely carry oxygen.
Yeah.
But as bad as a missense mutation is, it still produces a full-length protein. Nonsense and frameshift mutations are worse, right? They completely obliterate the structure.
Yes, a nonsense mutation accidentally creates a premature stop codon. The mnemonic is stop the nonsense. A ribosome encounters the signal and immediately terminates translation, releasing this truncated, structurally unstable protein fragment that usually just gets degraded.
But a frameshift is arguably the most destructive.
Because the code is read in discrete blocks of 3. If you delete or insert 1 or 2 nucleotides, any number not divisible by 3, you shift the entire reading frame for the rest of the gene. Every single amino acid downstream from the mutation site is completely scrambled.
The notes use Duchenne muscular dystrophy as an example for this.
It's a classic example. A frameshift mutation in the massive dystrophin gene results in a completely non-functional protein. And without dystrophin tethering the muscle fibers to the extracellular matrix, the muscle cells literally tear themselves apart during contraction. It leads to progressive, fatal muscle wasting.
The precision required to read and copy this code is just staggering. Which brings us to the replication process itself, and specifically this massive problem with the ends of our chromosomes. Linear DNA has a built-in flaw, doesn't it?
It does. It's known as the end replication problem. Every time the cell divides, the DNA polymerase cannot fully copy the very tips of the lagging strand, so the chromosomes physically shorten with every single division.
And to prevent the loss of critical genetic information, the ends of our chromosomes are capped with thousands of repeats of a non-coding sequence, right? TTGG.
Those are the telomeres. They act like the little plastic aglets on the ends of your shoelaces, providing this buffer zone of sacrificial DNA.
Eventually, even that buffer runs out. The telomeres get too short, the cell senses DNA damage, and it permanently halts division. It enters senescence.
That's the Hayflick limit. It is the molecular clock of cellular aging.
But the notes highlight an enzyme that can reverse this clock. Telomerase. How does that work?
Telomerase is a fascinating ribonucleoprotein complex. It actually contains its own internal RNA template. It physically binds to the shortened telomere and reverse transcribes new TT-TAGI repeats back onto the end of the chromosome.
Tagging it for greatness and glory, as the mnemonic goes.
Exactly. It lengthens the telomere and resets the cellular clock.
Okay, wait, I have a major question here. If we possess the biochemical machinery to literally reverse the molecular clock of aging, why is telomerase only active in our stem cells and germ cells? Why don't all our normal somatic cells just continuously express telomerase to maintain perfect tissue regeneration forever? Why do we age if the cure is already inside us?
Because endless, unregulated cellular regeneration is the literal definition of cancer.
Oh.
Yeah. The Hayflick limit is an evolutionary failsafe. If a regular cell accumulates dangerous mutations over time, we want it to hit that telomeric wall and die before it can grow into a tumor.
So the aging clock is actually a built-in kill switch.
It's an anti-tumor mechanism. We trade eventual systemic aging for immediate protection against rampant malignancy. And we know this is true because when cells do become cancerous, one of the primary mutations they absolutely must acquire to survive is the reactivation of telomerase. Over 85% of human cancers aggressively upregulate telomerase to achieve immortality.
Wow. So if you forcibly turned on telomerase in every cell, you wouldn't live forever. You'd just succumb to massive tumors very quickly.
Precisely.
That perfectly bridges us into our second major realm, genetics and inheritance. Because this beautifully complex replication process isn't just for keeping our own cells alive, it's how we pass the blueprint to the next generation.
Right.
But inheritance in a real clinical setting is rarely as clean as a simple dominant or recessive trait. We're dealing with
In the lungs, the epithelial cells use that CFTR channel to secrete chloride out into the airway lumen. Sodium ions naturally follow the chloride to maintain electrical neutrality, and then water follows the sodium via osmosis.
so the cell is pumping out salt water.
Yes, and this secreted water hydrates the mucus lining the lungs, keeping it thin and fluid so the cilia can constantly sweep bacteria and debris out of the airway.
So without the chloride channel, the chloride stays trapped inside the cell. The sodium stays inside. The water stays inside.
And the mucus in the airway becomes incredibly thick, dehydrated, and viscous. It literally crushes the cilia. The mucociliary escalator fails entirely. Bacteria become trapped in the stagnant sludge, leading to chronic, life-threatening pulmonary infections.
And the exact same osmotic failure happens in the pancreas.
It does. The pancreas secretes digestive enzymes through tiny ducts. Without CFTR, the exocrine secretions become so thick that they physically obstruct those pancreatic ducts.
So the enzymes can't get out to digest the food.
Worse, the digestive enzymes back up, prematurely activate, and literally auto-digest the pancreas itself, leading to massive fibrosis and severe fat malabsorption.
Oh, that's horrific. And in the reproductive system.
In males, the same thickened secretions completely block and destroy the vas deferens during fetal development, leading to infertility. So one broken chloride gate leads to 3 catastrophic, seemingly unrelated systemic failures. That is pleiotropy.
That structural perspective clarifies so much. Now let's unravel 2 terms that are frequently conflated in genetics: variable expressivity and incomplete penetrance.
Let's start with variable expressivity. This means that everyone who possesses the disease-causing genotype absolutely displays the disease phenotype, but the clinical severity varies astronomically between individuals.
Like a volume knob.
Exactly. Neurofibromatosis type 1 is a classic example. It's caused by a mutation in the NF1 gene, which codes for a negative regulator of the RAS signaling pathway.
So it's essentially a tumor suppressor gene for neural crest cells.
Correct. Now, take 2 patients with the exact same NF1 mutation. One patient might live a completely normal life, presenting only with a few flat, pigmented birthmarks, café-au-lait but the other patient might develop massive, disfiguring, benign tumors growing along every peripheral nerve in their body, alongside severe skeletal issues. Both patients absolutely express the disease, but the intensity is highly variable.
Got it. The volume knob is turned up to a 2 for one person and a 10 for the other. But what about incomplete penetrance?
Incomplete penetrance is not a volume knob, it is an on-off switch. Penetrance is a statistical calculation. Of all the people who have the mutated genotype, what percentage actually show any sign of the disease at all?
The notes use BRCA1 mutations for this.
Yes. BRCA1 is a tumor suppressor gene heavily involved in repairing double-strand DNA breaks. If a woman inherits a mutated BRCA1 allele, she's an extraordinarily high risk of developing breast or ovarian cancer. However, the penetrance is not 100%. She might live to be 90 years old and never develop a tumor.
Wait, why? If she has the broken gene, why wouldn't she get the cancer?
Because of the 2-hit hypothesis. She inherited one broken copy of BRCA1 from a parent that's the first hit. But she still has a second, perfectly functional copy of BRCA1 on her other chromosome. That single good copy is enough to maintain DNA repair.
Okay, so what has to happen for the cancer to start?
For the cancer phenotype to penetrate, she must acquire a random somatic mutation in that specific second copy of the gene within a specific breast or ovarian cell. That's the second hit. If that second random mutation never occurs in her lifetime, the disease never penetrates.
It really is a genetic Russian roulette.
Mm-hmm.
But there is a phenomenon detailed in the sources that feels almost malevolent in its mechanism: anticipation.
Yeah.
The idea that a genetic disease gets progressively worse, striking earlier and with more severity in every subsequent generation, like a genetic photocopy that gets blurrier and worse every time you copy it.
Anticipation primarily occurs in trinucleotide repeat expansion disorders. These are regions of the genome that consist of repetitive 3-letter stutters. For example, CAG, CAG, CAG.
Wait. If the DNA polymerase is essentially just a molecular copy machine, why does it suddenly start adding more repeats? Why doesn't it ever accidentally delete repeats and cure the lineage? Why does it only slip in one direction?
It has to do with the physical biophysics of single-stranded DNA during replication. As the DNA unwinds to be copied, these highly repetitive sequences are prone to forming secondary structures, like hairpin loops on the newly synthesized strand.
So the DNA is folding back on itself.
Right. The DNA polymerase is chugging along, but the new strand forms a loop behind it, physically pulling the polymerase backward. The polymerase loses its place, it slips, and it recopies a section of the repeats it literally just finished.
Wow. So the newly synthesized strand is physically longer than the template it was reading from.
Exactly. Take Huntington's disease, where the repeat is CAG, which codes for the amino acid glutamine. A normal individual has perhaps 15 to 20 glutamine repeats, but if that polymerase slips during germ cell formation, a parent might pass on 40 repeats.
Which is the threshold for the disease.
Yes. 40 repeats cause the resulting protein to misfold, aggregate in the neurons at the basal ganglia, and cause chorea and severe dementia, usually onsetting in the patient's 50s.
But then when that patient has kids,
Exactly. When they produce sperm or eggs, the polymerase slips again on this already unstable track. The child inherits 60 repeats. The expanded protein is far more toxic, aggregates much faster, and the child develops profound neurological symptoms in their 30s. The grandchild might inherit 80 repeats and develop the juvenile form. The disease anticipates the next generation.
the structural logic of that is horrifying, honestly. Let's keep exploring the nuances of inheritance. Imprinting. This concept really shatters the idea that maternal and maternal alleles are perfectly equal. Imprinting proves that a chromosome maintains an epigenetic memory of which parent it came from.
And this memory is established via massive DNA methylation during gametogenesis, the formation of the sperm or the egg. For the vast majority of our genome, both copies of a gene are active, but for a select few imprinted genes, the body deliberately, permanently methylates and silences one parent's copy. You rely entirely on the allele from the other parent.
And the devastating consequences of this are beautifully or tragically illustrated on chromosome 15. The sources contrast Prader-Willi syndrome and Angelman syndrome. Let's walk through the exact mechanism here, because both of these syndromes can be caused by the exact same microdeletion on chromosome 15. The physical DNA missing is identical, yet the clinical presentation is completely different based on the parent of origin.
Right. Let's examine the Prader-Willi region of chromosome 15 first. Evolutionarily, for this specific cluster of genes, the maternal alleles are heavily methylated and completely silenced. The body relies 100% on the active paternal alleles to produce these necessary proteins.
So the mother's copy is essentially turned off by default.
Correct. Now, if a child inherits a chromosome 15 from their father that has a microdeletion in this specific region, they have a catastrophic problem. The father's active copy is physically gone, and the mother's copy is epigenetically silenced. They produce absolutely zero protein from this gene cluster.
Which results in Prader-Willi syndrome. And the clinical hallmark there is hyperphagia, this uncontrollable, neurologically driven, insatiable appetite leading to rapid, severe obesity.
Now, look at the UBE3A gene, which is located in the exact same region of chromosome 15. For this specific gene, the imprinting is entirely reversed. The paternal copy is naturally methylated and silenced. The brain relies exclusively on the maternal copy of UBE3A to function properly.
So if the child inherits that exact same microdeletion, but this time they inherited the broken chromosome from their mother,
The maternal copy is physically gone, the paternal copy is silenced, and the child develops Angelman syndrome. This presents with severe intellectual disability, ataxia, and unprovoked frequent outbursts of laughter. It's a profound realization that the origin of the chromosome, the epigenetic tag placed on it in the testes versus the ovaries, dictates the pathology.
it really is. Okay, let's pull the scope back from individual families to the population level.
Yeah.
Population genetics. Specifically, the Hardy-Weinberg principle. Now, the math is often taught rote, p squared plus 2pq plus q squared equals 1. But the actual utility of this equation is figuring out why lethal genetic diseases haven't been completely eradicated by natural selection.
Exactly. It provides a mathematical framework for calculating the frequency of hidden carriers in a population.
Let's use cystic fibrosis again. It's a recessive disease, meaning you need two broken alleles, that's our q squared, to show symptoms. If the prevalence of CF in a specific population is roughly 1 in 3,200, that is q squared.
And by taking the square root of 13,200, we find Q, the frequency of the mutant CFTR allele, in the general gene pool, which is roughly 0.017 or 1.7%.
And since there are only two options, the normal allele P and the mutant allele Q, They must add up to 100%. So P is 1 minus 0.017, which is 0.983. The normal allele is roughly 98.3% of the pool.
And here is the clinical power of the equation. To find the carriers, the completely healthy people who possess one normal allele and one mutant allele without knowing it, we calculate 2PQ. 2 times 0.983 times 0.017.
Which comes out to a roughly 0.033, meaning over 3% of the population, or about 1 in 30 people, are walking around carrying a lethal cystic fibrosis mutation. But here's my question. If cystic fibrosis is so devastating, why does the allele frequency remain so high? Shouldn't natural selection have ground that down to 0 over thousands of years?
It's a phenomenon called heterozygote advantage. While having 2 copies of the mutant gene causes lethal cystic fibrosis, having just 1 mutant copy, being a carrier, historically conferred a survival advantage against cholera or severe secretory diarrheas.
Oh, because the broken chloride channel actually helps them retain water.
Exactly. The slightly impaired chloride secretion in carriers made them less susceptible to the massive fluid loss caused by the cholera toxin. The lethal trait is preserved in the population because the carriers survived pandemics. It's the same reason the sickle cell trait confers resistance to malaria.
It's an evolutionary trade-off. We tolerate a certain percentage of lethal recessive diseases in the population to protect the massive carrier base from infectious extinction. But the notes also mention that allele frequencies can change purely by accident, right? Entirely divorced from natural selection. The bottleneck effect.
Which is an extreme form of genetic drift. Imagine a large, diverse population. A sudden, catastrophic event, a massive volcanic eruption, a severe famine wipes out 90% of the population. The survivors do not perfectly represent the genetic diversity of the original group. Just by random chance, one of the survivors might carry a very rare recessive mutation.
And because the new breeding population is so incredibly small, that rare mutation is suddenly highly represented.
Exactly. When the population expands again, that once rare allele is now everywhere. This is the founder effect. It's why we see extraordinarily high rates of specific genetic diseases in isolated populations.
Yeah.
Like Tay-Sachs disease in Ashkenazi Jewish populations. It wasn't selection, it was a statistical bottleneck.
Wow. Okay, so we have built the DNA, we have unspooled the chromatin, we have inherited the alleles, and survived the bottlenecks. But this entire beautifully inherited genetic blueprint is completely inert on its own. DNA doesn't actually do anything. It's just an incredibly dense hard drive. To execute the code, to run the enzymatic machinery, the body has to import physical matter from the outside world. Which brings us to segment 3, nutrition.
The essential cofactors. We have built the highly complex protein enzymes, but many of these enzymes are synthesized in an inactive state. They require a specific non-protein molecule, a vitamin or a mineral, to physically bind to them, altering their three-dimensional conformation to allow substrate binding. Without these exogenous spark plugs, the chemical pathways of life grind to an absolute halt.
Let's start with the grand division. Fat-soluble versus water-soluble vitamins. The water-soluble ones, the B-complex and vitamin C, circulate freely in the blood and are rapidly excreted in the urine. We need a constant dietary supply. But the fat-soluble vitamins A, D, E, and K are entirely different beasts. They require bile acids and pancreatic enzymes to form micelles in the gut just to be absorbed.
And because they are hydrophobic, they don't dissolve in the blood. They are packaged into chylomicrons and ultimately stored in the massive lipid droplets of the liver and adipose tissue. This has 2 major clinical implications. First, if you have a fat malabsorption syndrome, like the cystic fibrosis we just discussed, you will rapidly become profoundly deficient in A, D, E, and K.
Right. And the second implication is toxicity, isn't it? So what does this all mean for someone taking massive supplement stacks? If the fat-soluble ones accumulate, couldn't you actually poison yourself?
You absolutely can. Taking mega doses of fat-soluble vitamins can literally poison you. The notes detail vitamin A or retinol specifically.
Let's talk about vitamin A.
It's a classic double-edged sword. At normal physiological levels, it's vital. It's an essential component of rhodopsin, the light-absorbing pigment in your retinas. A deficiency leads to severe night blindness. It is also required to maintain the proper differentiation of specialized epithelial cells.
Without it, the eye gets severely damaged, right? Bitter spots?
Yes. The delicate conjunctiva undergoes pathological keratinization, ultimately causing permanent corneal blindness. But if you take unregulated, massive supplement stacks...
Or eat polar bear liver, which is famously toxic.
Right, because of its massive retinol content. The toxicity is profound. Retinoic acid is not just a coenzyme, it acts as a steroid hormone. It passes directly into the nucleus and acts as a transcription factor, forcing genes open or closed. Acute toxicity causes severe intracranial hypertension, presenting as massive headaches. Chronic toxicity causes bone pain and liver damage.
And the notes give a very stark warning regarding pregnancy.
Because retinoic acid directly modulates the expression of homeobox genes, the master regulator genes that dictate the spatial layout of the developing embryo. If a pregnant woman has massive levels of vitamin A, it completely scrambles the spatial patterning of the fetus. It is highly teratogenic, causing severe cleft palates and cardiac abnormalities.
Which is why acne medications like isotretinoin require sort of rigorous pregnancy prevention protocols.
Yeah.
Context and dosage are everything. Let's move to the gears of the energy engine, the B complex. These water-soluble vitamins are the literal molecular wrenches for our enzymes. Let's look at B1, thiamine.
Thiamine is converted in the body to thiamine pyrophosphate, or TPP. TPP is the mandatory prosthetic group for several massive dehydrogenase enzyme complexes. The most critical is a pyruvate dehydrogenase complex,
Which is the toll gate between glycolysis in the cytoplasm and the TCA cycle in the mitochondria. Pyruvate dehydrogenase takes the 3-carbon pyruvate molecule from glucose and strips off a carbon to create acetyl-CoA.
and the enzyme physically cannot perform that decarboxylation without TPP stabilizing the reaction. If you lack thiamine, pyruvate dehydrogenase shuts down. Pyruvate cannot enter the mitochondria. Instead, it backs up and is shunted into lactic acid.
So the cell's main energy grid blacks out, and it's forced onto emergency backup generators, producing minimal energy and a ton of acid. And the brain and heart fail first.
Which leads to beriberi, characterized by profound cardiac failure. But in Western medicine, we most frequently see thiamine deficiency in the context of chronic alcohol use disorder. Alcohol directly inhibits the transport proteins that absorb thiamine in the gut. This leads to Wernicke-Korsakoff syndrome.
The notes mention Wernicke encephalopathy as the acute phase, confusion, ataxia, and ophthalmoplegia, which is paralysis of the eye muscles.
Because the highly active metabolic regions of the brainstem are literally starving for ATP and drowning in lactic acid. If untreated, it progresses to Korsakoff syndrome, where the continuous energy failure causes permanent necrosis of the mammillary bodies in the brain. The patient develops profound amnesia and confabulation, inventing elaborate false stories to fill the gaping voids in their memory.
Now, there is a massive life-or-death clinical pearl here from the text. If a severely malnourished chronic alcoholic patient is brought into the ER, the instinct is to give them an IV bag of dextrose to give them energy. But the notes say if you do that without giving thiamine first, you will actively kill them. Why?
Because you are suddenly flooding a system with glucose, completely unaware that the cellular machinery to process that glucose is broken. The cells aggressively break the dextrose down into pyruvate, but without thiamine, the pyruvate dehydrogenase toll gate is locked shut. The massive wave of pyruvate rapidly converts to lactic acid. You instantly drain whatever minuscule cellular ATP reserves were left, precipitating a catastrophic, fatal exacerbation of Wernicke encephalopathy.
Wow. You have to give the thiamine first. It's like flooding a car engine with gasoline when the spark plugs have been removed. You just destroy the engine. Okay, B3 niacin, the precursor to NAD+ and NADP+ numer.
The ultimate electron carriers. Niacin is incorporated into NAD plus ADA, which acts as the primary acceptor of hydride ions during the oxidation of food molecules. Without niacin, you cannot extract electrons from your food.
And a severe deficiency causes pellagra. The mnemonic is the 3 Ds: diarrhea, dementia, and dermatitis. But the dermatitis is very specific. It forms a hyperpigmented rash on sun-exposed areas, classically presenting as a broad collar around the neck known as the Casal necklace.
And what's chemically fascinating is that the body can synthesize a small amount of niacin endogenously, but it has to use the essential amino acid tryptophan as the starting material. Therefore, any pathology that disrupts tryptophan absorption will induce pellagra.
Like carcinoid syndrome.
Yes. A neuroendocrine carcinoid tumor secretes massive amounts of serotonin. Serotonin is also synthesized from tryptophan. The tumor acts as a metabolic sink, stealing all the body's available tryptophan to make serotonin, leaving none for niacin synthesis. The patient develops pellagra secondary to the tumor.
The systemic interconnectivity is just incredible. Moving to the final 2 B vitamins, B9, which is folate, and B12, cobalamin. These two are inextricably linked, and both are absolutely required for the synthesis of DNA.
Specifically, they are involved in 1-carbon metabolism. They transfer single methyl groups to build the purine and pyrimidine nucleotides. For instance, you need a folate derivative to convert DUMP into DTMP, the thymine building block of DNA.
So if you lack folate or B12, you can't build thymine. If you can't build thymine, you can't replicate DNA. And the cells that divide the fastest in the human body, the red blood cells in the bone marrow, are the first to crash.
They attempt to divide, they synthesize massive amounts of RNA and protein for the cytoplasm, but the nucleus is paralyzed due to the lack of DNA nucleotides. This results in enormous, structurally unstable red blood cells, a classic megaloblastic anemia.
But there's a vital clinical distinction between folate and B12 deficiency. Folate deficiency causes the anemia. B12 deficiency causes the anemia and destroys your spinal cord.
Through a completely separate pathway. B12 is the mandatory coenzyme for an enzyme critical for the metabolism of odd-chain fatty acids. Without B12, methylmalonic acid accumulates to highly toxic levels. This abnormal fatty acid gets erroneously incorporated into the lipid myelin sheaths that insulate the tracts of the spinal cord,
The insulation literally degrades from the inside out.
leading to subacute combined degeneration. The dorsal columns fail, causing a loss of proprioception. The lateral corticospinal tracts fail, causing spastic paresis. It is profound, permanent neurological destruction.
And B12 is incredibly complex to absorb, requiring intrinsic factor from the stomach. But here's the terrifying trap mentioned in the notes. What happens if a patient has a B12 deficiency, but they take a massive folate supplement instead?
This is a classic board question. The massive influx of folate will forcefully bypass the metabolic block, allowing thymine synthesis to resume. The anemia will miraculously resolve. The patient's blood work will look perfectly normal, but the folate does absolutely nothing for the methylmalonic acid pathway. The subacute combined degeneration of the spinal cord will continue to aggressively, silently destroy the patient's nervous system. It is a catastrophic clinical error to treat a megaloblastic anemia with folate without confirming B12 levels first.
That is terrifying. Okay, wrapping up the vitamins, we have to talk about structural integrity. Vitamin C, ascorbic acid. We all know the pirate stories of scurvy, but what is actually happening at the molecular level when you deprive a human of vitamin C?
It comes down to collagen. Collagen is a structural protein. It forms a massive triple helix scaffolding that supports your skin, blood vessels, gums, and bones. To form that incredibly tight rope-like helix, the collagen strands must be heavily cross-linked, and that requires specific amino acids proline and lysine to be hydroxylated.
Meaning an OH group is grafted onto them.
Exactly. The enzymes that do this require an iron atom at their active site to function. During the reaction, that iron atom is oxidized from a 2+ state to a 3+ state. Once it hits the 3+ state, the enzyme is completely dead.
So where does vitamin C come in?
Vitamin C acts as a dedicated reducing agent. Its sole job in this pathway is to physically donate electrons to that iron atom, constantly reducing it back to the active 2+ state, keeping the hydroxylase enzymes firing.
So without vitamin C in your diet, the iron oxidizes, the enzymes die, you stop hydroxylating your collagen, and your newly synthesized collagen cannot cross-link into a triple helix. The entire structural scaffolding of your body becomes a weak, unraveled mess.
Which is the mechanical definition of scurvy. The weak collagen causes the capillary walls to easily rupture, leading to massive petechial hemorrhages. The periodontal ligaments disintegrate, so the teeth literally fall out. The hair follicles hemorrhage. The structural integrity of the human body dissolves.
It sounds identical to the genetic collagen disorders mentioned, like Ehlers-Danlos syndrome, where a mutation in the collagen gene itself causes hyperflexible joints.
The end phenotypic result is identical. But one is a failure of the genetic blueprint, and the other is a failure to supply the necessary exogenous electron donor to the manufacturing plant. It also highlights why taking vitamin C with an iron supplement dramatically increases the iron's absorption in the gut. It keeps the dietary iron in that absorbable 2+ state.
Unless you have hemochromatosis, the genetic iron overload disease, in which case absorbing extra iron is incredibly toxic. Again, molecular context is everything. Which brings us to the grand finale. Segment 4. Metabolism, the engine of life. We understand the DNA blueprint, the protein machinery, and the required vitamins. How does this machinery actually extract kinetic energy from the chemical bonds of a carbohydrate?
We must trace the highly orchestrated disassembly line of carbon. The goal is to systematically strip high-energy electrons off of the food molecule and use their energy to force a phosphate group onto an ADP molecule, generating ATP, the universal high-energy currency of the cell.
It starts in the cytoplasm with glycolysis. The cell takes a 6-carbon glucose molecule and, through 10 steps, cracks it in half into 2 carbon pyruvate molecules. This doesn't require oxygen, but the energy yield is pathetic, right?
Very pathetic. It costs 2 ATP to start, and you only get 4 back. A net gain of 2 ATP per glucose molecule. It's ancient, rapid, and inefficient. To unlock the true energetic potential, the pyruvate must be transported into the mitochondrial matrix. There, it's converted to acetyl-CoA, which enters the tricarboxylic acid cycle, the TCA or Krebs cycle.
Now, I always found the TCA cycle frustrating to learn because it doesn't actually produce much ATP either. It just spins around, generating a single molecule of GTP per turn and blowing off the carbon as CO2. What is the actual point?
The point of the TCA cycle is not to make ATP directly. It is an electron harvesting machine. As the carbon bonds are systematically broken apart and exhaled as carbon dioxide, the cycle captures the released high-energy electrons and transfers them to specific carrier molecules, NAD+ and FAD, reducing them to NADH and FADH2.
So they're essentially heavy-duty molecular batteries that have just been fully charged.
Precisely. The TCA cycle's job is to charge the batteries. And those massive electron-laden batteries are immediately shuttled to the inner mitochondrial membrane, where the true magic happens. Oxidative phosphorylation and the electron transport chain.
This is the chemiosmotic theory, which is arguably one of the most brilliant mechanisms in all of biology.
Yeah.
Walk us through how the transfer of an electron actually creates ATP.
Embedded within the inner mitochondrial membrane are four massive protein complexes. NADH and FADH2 dock at the first complexes and hand over their high-energy electrons. As these electrons are passed like a hot potato down the chain, they drop to lower and lower energy states.
And the protein complexes use that released energy to do physical work.
Yes, they act as proton pumps. They use the energy from the passing electrons to physically pump hydrogen ions, protons, out of the mitochondrial matrix and into the tiny inner membrane space.
They are pumping positively charged protons against their concentration gradient, packing them into a microscopic space. It's the exact equivalent of a hydroelectric dam pumping millions of gallons of water up into a high-altitude reservoir.
The potential energy is staggering. The intermembrane space becomes highly acidic and highly positively charged. The protons desperately want to flow back down their gradient to equalize the pressure, but the inner mitochondrial membrane is strictly impermeable to them.
There's only one way back in.
A single, highly specialized protein channel called ATP synthase. It is literally a microscopic molecular motor. As the protons violently rush back into the matrix through ATP synthase, the physical force of their movement causes the central stalk of the enzyme to physically rotate at thousands of revolutions per minute.
It is a literal microscopic turbine. Think about that as the central bank printing the big bills.
And as that turbine spins, it forces conformational changes in the catalytic head of the enzyme, slamming ADP and a free phosphate group together with such mechanical force that it forms the high-energy bond of ATP, up to 32 molecules of ATP per original glucose molecule.
That's the payout. And what happens to the electrons?
To keep the chain moving, those spent low-energy electrons at the end of the line are handed off to a molecule of oxygen, which picks up 2 protons to become water. Oxygen is the terminal electron acceptor. That is the fundamental reason you breathe.
That is structural biology at its finest. But this delicate chemiosmotic gradient raises a terrifying vulnerability. What happens if the inner mitochondrial membrane springs a leak? What if the dam breaks and the protons can bypass the ATP synthase turbine?
That phenomenon is called uncoupling. There are chemical agents that are highly lipophilic and can easily carry protons directly across the inner membrane. The protons rush back into the matrix, completely destroying the electrochemical gradients.
Well, the turbine stops spinning. No ATP is produced.
None. But the cell immediately senses the massive drop in ATP. In a desperate panic to restore the gradient, the electron transport chain goes into overdrive. It accelerates the TCA cycle, rapidly burning through all available glucose and fat stores, furiously pumping protons out, only for them to immediately leak back in.
The engine is redlining, but the transmission is disconnected.
And the thermodynamic consequence is severe. The massive amount of potential energy from the rapidly burning fuel is no longer being captured in the chemical bonds of ATP. That energy must go somewhere. It is released entirely as pure heat.
The notes highlight a very common, very dangerous clinical scenario of this: aspirin overdose.
In massive toxic doses, salicylates act as uncoupling agents. The patient presents to the ER with an incredibly high, life-threatening fever hyperthermia, but they also have severe metabolic acidosis and are hyperventilating. They are breathing rapidly because their electron transport chain is consuming oxygen at an astronomical rate trying to fix the leak. They are literally burning to death from the inside out, while their cells simultaneously starve for ATP.
It's the ultimate metabolic short circuit. But wait, isn't there a physiological deliberate use for this? What about hibernating animals?
A brilliant evolutionary adaptation. Brown adipose tissue, found heavily in hibernating mammals and human infants, possesses a protein called thermogenin. It is a deliberate, highly regulated, uncoupling channel. The organism purposefully sacrifices ATP production to generate massive amounts of metabolic heat, keeping their core temperature stable during freezing conditions.
It turns a toxic mechanism into a survival tool.
Yep.
Let's look at another survival tool. What happens to the metabolic engine when the fuel simply stops arriving? The bagel has been processed, the ATP is synthesized, but now it has been 18 hours and we are in a fasting state.
The hormonal milieu shifts drastically. Insulin plummets and glucagon and epinephrine rise. The body has one singular priority. The brain and the red blood cells require a constant, unyielding supply of blood glucose. The liver becomes the altruistic organ, sacrificing its own energy stores to maintain systemic blood sugar.
The liver enacts phase 1, glycogenolysis. It breaks down its massive, highly branched storage polymer of sugar glycogen.
Glucagon binds to receptors on the liver cells, initiating a cascade that ultimately activates glycogen phosphorylase. This enzyme rapidly cleaves single glucose molecules off the branches of glycogen, releasing them into the bloodstream.
But the liver's glycogen stores are finite. They run out in about 24 hours. Then we enter phase 2.
Gluconeogenesis. The liver physically synthesizes brand new glucose molecules from scratch. It takes glycerol from broken down fats, lactate from exercising muscle, and heavily cannibalizes amino acids from muscle tissue, running the glycolysis pathway in reverse to force the production of new glucose.
And if the starvation continues for days or weeks, we can't just cannibalize our skeletal muscle forever, or we'd lose the ability to breathe.
Exactly. To spare the muscle proteins, the body shifts entirely to lipid metabolism. Adipose tissue dumps massive quantities of free fatty acids into the blood. The liver processes them into ketone bodies, acetoacetate and beta-hydroxybutyrate. These water-soluble lipid derivatives cross the blood-brain barrier. The brain adapts its machinery to burn ketone bodies for ATP, drastically reducing its demand for glucose and sparing the muscle tissue.
It is an incredibly sophisticated triage hierarchy. But, like every pathway we've discussed, genetic mutations can completely break the system. Let's look at the glycogen storage diseases. The notes focus on von Guericke disease.
Von Guericke, or type I glycogen storage disease, is caused by a deficiency in glucose 6-phosphatase. This is the absolute final enzyme in the liver's glucose export pathway.
So the liver can break down glycogen perfectly fine. It can even run gluconeogenesis perfectly fine.
Yes, it successfully creates glucose 6-phosphate. But because it lacks that final phosphatase enzyme to remove the phosphate group, the glucose is trapped. It cannot cross the cell membrane to enter the blood. The exit door is permanently locked shut.
The physiological consequences must be devastating.
They present with profound, life-threatening fasting hypoglycemia. Because the liver cannot release sugar between meals, the brain is constantly starved. Simultaneously, the trapped glucose 6-phosphate backs it up, stimulating massive, continuous glycogen synthesis. The liver swells to enormous proportions. To survive, these patients must consume raw, uncooked cornstarch every few hours, day and night, to provide a slow, constant trickle of intestinal glucose.
The system works perfectly right up until the final step. And the notes also mention Pompe disease type 2.
Pompe is arguably more tragic. A tiny amount of glycogen is normally degraded inside the lysosomes by an enzyme called lysosomal alpha-1 glucosidase. In Pompe disease, this enzyme is defective. The glycogen builds up inside the lysosomes, physically destroying the architecture of muscle tissue, most notably the heart.
Pompe crashes the pump.
Yes. It leads to massive cardiomegaly, severe hypotonia, and typically fatal heart failure in early infancy. The engine chokes on its own fuel reserves.
Let's hit the final piece of the metabolic puzzle. You mentioned the adipose tissue dumping fatty acids into the blood. But lipids are hydrophobic. Blood is a watery environment. How do massive quantities of fat traverse the circulatory system without instantly separating like the oil and vinegar in a salad dressing?
They require sophisticated transport vehicles known as lipoproteins. These are micellar spheres. The outer shell is composed of water-loving phospholipids, and the core is densely packed with completely hydrophobic triglycerides and cholesterol.
And embedded in that outer shell are apolipoproteins. These aren't just structural. They are the molecular ZIP codes that tell the lipoprotein where to go.
Precisely. When you eat dietary fat, your intestines package it into massive cargo ships called chylomicrons. They deliver free fatty acids to the tissues, and the remnants return to the liver.
And when the liver manufactures its own fat from excess carbohydrates, it packages them into VLDL. As VLDL drops off its triglycerides, it becomes denser, eventually transforming into the infamous low-density lipoprotein, LDL.
LDL is the primary cholesterol delivery vehicle, but it is notoriously pathogenic. If LDL levels are too high, the particles penetrate the delicate lining of the arteries. They become oxidized, engulfed by macrophages, which transform into massive foam cells and initiate the inflammatory cascade that builds atherosclerotic plaques. LDL is lethal. It drives coronary artery disease.
Which is why we rely on HDL, high-density lipoprotein.
HDL performs reverse cholesterol transport. It acts as a molecular vacuum cleaner, extracting excess cholesterol from the peripheral cells and transporting it directly back to the liver for excretion. HDL is healthy.
The ultimate goal of modern cardiology is to lower that LDL, and the sources detail an absolutely fascinating pharmacological mechanism for this involving PCSK9. We know the liver naturally clears LDL from the blood using specific LDL receptors on its surface. The receptor grabs the LDL particle, pulls it into the cell, and destroys it.
Yes. But to be efficient, the liver cell normally recycles that LDL receptor back to the surface. However, there is an endogenous protein called PCSK9. When PCSK9 binds to the LDL receptor, instead of being recycled, the receptor is marked for lysosomal degradation. It is permanently destroyed.
So PCSK9 actively destroys the liver's ability to pull LDL out of the blood.
Correct. The more PCSK9 you have, the fewer LDL receptors you have, and the higher your blood LDL levels soar.
It seems like an evolutionary flaw, but it allowed pharmacologists to design PCSK9 inhibitors antibodies that neutralize the PCSK9 protein in the blood.
It is a masterstroke of applied biochemistry. By injecting a PCSK9 inhibitor, you prevent the destruction of the LDL receptors. The liver retains massive numbers of receptors on its surface, continuously pulling LDL out of circulation. The blood levels of LDL plummet, dramatically halting the progression of atherosclerosis.
It is the perfect synthesis of understanding the molecular mechanism to physically alter the macroscopic outcome. And that brings our entire journey full circle. We started deep inside the nucleus, looking at the electrostatic physics of looping DNA into chromatin. We navigated the volatile, stuttering replication forks of inheritance. We introduced the critical exogenous vitamins required to activate the proteins. And finally, we watched the mitochondrial turbines turn those nutrients into kinetic, crawling life.
The human body is a deeply interdependent, highly fragile web of thermodynamics. A mutation in a single nucleotide, a deficiency of a single vitamin, or the malfunction of a single microscopic receptor ultimately cascades into the macroscopic failure of the entire organism.
Which leaves us with one final profound connection that binds this entire biological framework together. We spent the first segment talking about epigenetics, how adding bulky methyl groups to the DNA sequence physically blocks the transcription machinery, acting as the ultimate off switch for your genetic blueprint.
DNA methylation.
But those methyl groups don't just magically appear out of the ether. They are physical carbon atoms. Where do they actually come from?
They are derived entirely from your metabolism and your diet. Specifically, the folate and B12 1-carbon metabolism pathways we discussed. Those vitamins are responsible for synthesizing a molecule called SAME.
And SAME is the universal methyl donor for the entire body. It is the molecule that physically hands the methyl group to the DNA methyltransferase enzymes.
Which means the physical food you consume and the specific vitamin coenzymes you absorb directly dictate the availability of the metabolic currency required to silence your genes. The state of your cellular metabolism physically dictates the availability of the chemical tags used to permanently alter your DNA architecture.
The environment and the blueprint are not separate entities. They are having a direct chemical conversation inside your cells right now. You are not just what you eat. Your actual genetic expression is dictated by what you eat. Your breakfast has the potential to alter the physical geometry of your chromatin and rewrite your cellular destiny.
It is the ultimate convergence of the cosmic and the biochemical.
So the next time you gaze up at the night sky and marvel at the fact that you are made of star stuff, take a moment to appreciate the gritty, terrifyingly complex, carbon-crawling machinery that keeps those atoms organized enough to even perceive the stars in the first place. Thank you for taking the plunge with us on this deep dive.